Our next talk is by PhD candidate Taylor Paisie from the University of Florida in the US. I haven't personally worked with Taylor, but I think that we're second research cousins through Taj Azarian. She's working on a topic really close to my heart, evolutionary dynamics of the seventh global pandemic wave of Vibrio cholera. So I appreciate that. Taylor brings I would say that she brings like a lot of fun to our community. Actually, she displayed this amazing Zoom background one time with her sitting on the Iron Throne, and she had a suggestion for the conference for us all to be in PJs, which my wife vetoed. Taylor's bio also says that she enjoys typical nerd activities such as Game of Thrones and playing video games, and so I'm sure that she could be a really fun teammate tonight for trivia night. I'm looking forward to hearing more from Taylor. Please join me in welcoming Taylor by clicking your Zoom applause icons. And Taylor, please take it away.
Hi, everyone. I'm Taylor. So let me get my screen up here and I'll start sharing. Sorry, I did it the wrong way. All right. Share. All right. Can everyone see?
Yep, we can see it.
All right, right screen, okay. So my name is Taylor, Taylor Paisie. I am from the University of Florida. I hopefully am on the last very leg of my PhD. So maybe the next time you see or talk to me, I will be Dr. Taylor Paisie. So hope for that. I am in Dr. Marco Salemi's lab at the University of Florida, the Emerging Pathogens Institute. And for the past couple years now, for my PhD project, I've been working on the genomic epidemiology of toxigenic Vibrio cholera 01, mainly in Haiti, but I've done some side projects with cholera in the, I always say Dominican Republic, I'm sorry if I do that, it's Democratic Republic of Congo. As I work with Haiti, I would hope some people would understand since they're on the same island. So some of the things I want to go over today are I'm going to go over genomic epidemiology and bacterial phylogenomics. Then my two projects that involve Haiti are clinical and environmental transmission of Vibrio cholera. And we saw a shift from the Ogawa to the Inaba serotype also in Haiti. And then I will briefly go over the project I've been working on in the DRC. So what is phylogenetics? It's the merging of immunodynamics, epidemiology, and phylogenetics. And this helps us better understand evolutionary processes of pathogens at different scales. We can, for example, we can track like the pathogen's emergence or look at the demographic history of a pathogen and changes in its effective, such as changes in its effective population sizes. So advances in next generation sequencing technology have allowed for these methods to be put onto for viral pathogens for a while now with much smaller genomes, but now we can start looking at bacterial pathogens with the advances in this technology. And the picture to the right shows just like the workflow of phylogenetics and what you go through. So I'm sure you've seen these pictures a million times, just looking over the costs and how it's gone down for years to come now. And that's why we were able to look at bacterial pathogens now. So what is genomic epidemiology? It's the use of whole genome sequencing for an outbreak investigation. Next generation sequencing advances allow us to do it with bacterial pathogens, and the extensive amounts of computational resources that have been on the rise also allow us to work with hundreds of bacterial genomes. And this, I would say, is the golden age of tracing outbreaks. Now, it's been quarantined for a while, so don't judge me, but I had a little fun on YouTube and I did the start. It was supposed to go with the slide. I don't know if everyone can hear it, but I had a little too much fun. Okay. So kind of what we do and what we did for our studies that I go through and I've been involved in is we take samples from the field do lab extract, like do DNA extraction. We do DNA sequencing, and then we can do different, we can do de novo assembly of these sequences or map to reference genomes, both. Do comparative genomics and look at the phylogenies and the relationships between these sequences. And then potentially, maybe if we have the right data, do inference of transmission. So in order to look at bacterial genomics, or to do this on cholera, for example, I had to make a reference-based reference mapping pipeline for my cholera samples that we collected. And in honor of John Snow, who is considered one of the founding fathers of modern epidemiology, I named my pipeline after him. And also, I said I was a fan of Game of Thrones after Jon Snow too, and I had a little fun. I found the biology of cholera. So I used this pipeline in order to do my phylogenetic with my projects that I'm going to go over. So a little introduction to cholera. Is caused by the bacterium Vibrio cholera. It's a severe acute dehydrating diarrheal disease, causes 1.4 to 4.3 million cases a year and 28,000 to 142,000 deaths a year. The symptoms include watery diarrhea, nausea and vomiting, dehydration, and this is a little, I didn't know this before, like looking into cholera and everything, 80% of the people infected with cholera are actually asymptomatic. And just a little fun, those are my giant microbes. And then I found a little cute animation. So the transmission of cholera is usually due to poor sanitation environments, so unsafe drinking water and food, poor hygiene, and it's spread by person to person, mainly by fecal contamination, and the highest risk areas are usually overpopulated urban areas. Another way that cholera can be transmitted, though, is environment to person. So Vibrio cholera can actually circulate in aquatic environments for quite a long time and infect humans. So what's this hypothesis? This is the clinical to environmental transmission hypothesis that we have. It's my baby Yoda telling you not to see aquatic animals might get cholera. Cute. So A is showing us if there's evolution in the aquatic environment of the bacteria. You will see in the tree green branches, which signifies environmental evolution. So you're seeing evolution in the environment, and then it infects humans, and then possibly causing another outbreak of humans. In the second tree, you're seeing mainly a transmission or a tree that is showing when the bacteria infects humans, but it's not showing the evolution in the environment. And as you can see, branches of the bacteria, the green, just die off. They're not ongoing evolution like we see in A. So if we're seeing environment, this is suggesting what we want to see if we're seeing an aquatic reservoir and evolution of the bacteria in the aquatic reservoir, we should see green in the trunk of our phylogenetic trees that we make. So why is Haiti a hotspot for infectious disease outbreaks? Well, you probably have heard there was, in January 2010, there was a catastrophic earthquake around Port-au-Prince, Haiti. And this just devastated already not very great healthcare infrastructure. And then leading to UN peacekeeping troops coming to Haiti and introducing Vibrio cholera in October 2010. Although this is terrible, it did give us the unique opportunity to study the emergence and spread of Vibrio cholera in Haiti, since it was a single clonal source introduction to a country that hadn't seen cholera in over 150 years. So the population was completely naive to the bacteria. And then this is where I have a map here showing where we collected samples mainly for our environmental samples, which is in the Gressier region of Haiti, where US has a lab there. So that's mainly why we work. So what we want to see in this clinical to environmental transmission, we want to assess the ongoing evolutionary changes in the clinical and evolutionary or environmental strains. Look on the genetic diversification that's driven primarily by the evolutionary changes accumulating during transmission. We want to look at the aquatic can the aquatic reservoirs sustain the bacteria evolution independently from the human host? So see those green internal trunks of the trees. Is the evolution of aquatic reservoirs driven by environmental stressors such as rainfall, water temperature, nutrient availability, phage population? So what we have here is, first we have a map of our geographical distribution of our environmental isolates that we collected and the clinical isolates we collected in Haiti from 2010 to 2015. And as you can see, we collected mainly in the Grazier region, but we had environmental sites. So then B is showing the number of monthly cases counts of cholera in the West Department from 2010 to 2015, so just in that department of Haiti. And then the blue is actually the average monthly rainfall. And as you can see, we see case counts kind of correlate with the monthly so when you have a peak in rainfall, you usually see a peak of cholera cases. So it's associated with the rainy season more, we notice, than water temperature or anything. And then C is actually just the ancestral state reconstruction that we used, using a maximum likelihood tree, and this was inferred using a program called PASTML. And as you can see, we see intermediates, we see purple's clinical represents clinical samples and green represents environmental samples. And you see intermediates of green. So you see purple to green to purple, and that is also indicative of environmental evolution leading to subsequent infections of humans and then outbreaks. So here we have a maximum clade credibility tree that we did phylogeography, which usually you do by location, but here instead we did it we assigned a state as clinical or environmental. And as you can see, we do see green suggesting in the trunk of the tree, suggesting ongoing environmental evolution. And then B is where you see B is the trunk rewards. Right here is the trunk rewards proportion. So this takes the ancestral location state estimated over time inferred using a Markov chain model. So, and as you can see around 2013, 2014, we do see it in that environmental state more than the clinical state, suggesting it's staying in that environmental state and with ongoing evolution. And so C is just the number of jumps in percentage of all the intrastate and interstate transitions. So interstate, clinical to clinical, environmental, and interstate, environmental to clinical to clinical to clinical. And we normalize this by total numbers of transitions, and this was obtained by our tree, the MCC phylogeny. Sorry. And so as you can see, and then, okay, let me restart. And then D is the weighted average of synonymous substitution rates and non-synonymous substitution rates for only environmental Vibrio cholera isolates and then only clinical Vibrio cholera isolates. And as you can see, we see a large number of a higher rate of non-synonymous mutations in our environmental only samples. suggesting that we may be having some positive selection going on. So here we have a evolution and adaptation of Vibrio cholera in the Haiti aquatic reservoir. So we just did a time phylogeny with only the environmental isolates, looking at the evolution and of those environmental isolates. And then here, marked by the boxes on the tree, are the snips that we detected along the trunk and that caused non-synonymous mutations. So as you can see, like 1, 2, if you see number 2 on the tree here, this gene EXEA in Vibrio cholera had a K, a non-synonymous mutation of amino acid K changing into a Q, and so on throughout the tree here as labeled. And then we have the map showing our sampling sites, and it's grouped by aquatic source, and they are labeled according to their region labeled here on the map. So as you can see, those mutations happening on the tree then were on from that sample on, suggesting these mutations were ongoing, being selected for in the aquatic environment. So here we did some mixed transmission modeling of cholera and vaccine prediction. So up top A, we have the affected population size observed from our data and in simulation with environmental replication, and affected population size in simulation without environmental replication. So as you can see with our above 1, without environmental, you do see a peak at a certain point, but it's going down, whereas the environmental is going back up again. And then for B, we have distinct scenarios of vaccination and control for the model simulation with environmental replication. In particular, vaccination coverages of 64%, and then we have 88% vaccination coverage, and then we have 64% with a 10% increase in environmental decay rate. So as you can see here, the 2 options that make the most sense and follow the same are 64% vaccination coverage and 10% increase in environmental decay rate, as well as the 88% vaccination coverage. So that 10% increase in environmental decay rate means taking some measures in the environment, such as chlorination or something like that, to kill the bacteria. So you either have to do some type of environmental intervention or do 88 vaccination coverage of the population in order to have a decrease in cases. So some of our conclusions for this. So we see evidence of aquatic reservoirs being established in Haiti. We see the evolution associated with the rainy season, not really due to water temperature. We see stressors in the environment that may be driving the evolution of new Vibrio cholera variants. And we kind of also see that blooms in the aquatic reservoir may be a good indicator for a potential outbreak, leading to a potential outbreak. And this may cause cholera to become endemic in certain areas. So this paper with this data just came out, and it was just published in PNAS. I have a little cool scanning code that'll take you to the link of the paper. So it just came out, and really proud of that work. So check it out. So another thing I want to go over is Vibrio cholera 01 serotypes. So over the past 5 years, we've seen in Haiti, the original introduced Ogawa serotype has shifted now to the Inaba serotype. So we want to assess the evolutionary dynamics that are causing this switch from the Ogawa to the Inaba serotype. This could be due to evade Ogawa-induced host immunity, but we see Inaba serotype went from a very low number of samples to almost all the samples we collected in Haiti starting in 2015. So here I have a global maximum likelihood tree. I just collected representative sequences from places around the world from NCBI, download them, include some of my sequences from Haiti, which are marked there. And I have them labeled by serotype on the end of the node with the circle. And then the color-coded regions are the location. And I have it just by region right there. And then we have labeled clade 1 and clade 2, which are represented B and C. And as you can see here, I did phylogeography here for these two, and I have them labeled by their location on the side and their serotype. So what I want to represent by this figure is to basically show when you see outbreaks of the Ogawa and the Inaba serotype, it's usually caused by a separate introduction into that region. So you're usually from where it's a source, like the Bay of Bengal, somewhere like there. So you see a lot of introductions from the Bay of Bengal to Africa or someplace like that. And that's what we want to represent, is you kind of see an intermixing showing that's caused by separate introductions. So here I have a figure showing where we collected our samples in Haiti and the percentage of the Inaba Ogawa isolates collected in various regions. So here's a bar chart of all the A is showing a bar chart of all the samples we collected and the percentage of them Ogawa Inaba isolates. So here you can see in 2015, we started to see an increase in the Inaba samples to almost all of them we collected. And then as time went on each year, starting in 2015, marked below, almost all the samples we collected were Inaba, showed by the bar charts in both regions where we collected the most samples, which were the Gressier and the Jacmel region. So here I have an MCC tree. And I did phylogeography again. And the states I did here, instead of clinical environmental, I did serotype. So I did labeled each sample by serotype, and I did the Ogawa to the Inaba serotype. So these boxes up here that are labeled 1, 2, 3, and 4 are showing Inaba samples and where their mutation in a gene called the WBET gene, which has been shown to cause the switch in serotype from Ogawa to Inaba, where the mutation in that gene is and what it does. So one here, for example, causes a premature stop codon, which is labeled right here with this black line. And so that premature stop codon then most likely leaded to a truncated WBET protein, which turned it to the Inaba serotype. Same for 2, you saw a premature stop codon. And labeled 3, you also saw a premature stop codon. So for the whole shaded area 4 here, though, we saw a premature stop codon in this location in the WBET gene in all of these samples. We see the same mutation in every sample in the gray. We saw a premature stop codon in position 305 of the gene. So here I just kind of have a figure with the WBET gene below, showing the location of these figures above, the nucleotide sequence and their corresponding amino acids. And so I picked these samples because we did an analysis that I'll go over next, showing where the premature stop codon in the WBET gene is located in these samples. Yes, so, sorry, is located in these samples, and they all are so this, the HC9 1961 and 2541 represents that shaded gray area in the figure before. So that represents all those samples, and they all have the same premature stop codon located in this position in the WBET gene. So here we did an experiment called a mass Western, and what we did is we took the WBET gene and we wanted to see if the protein was expressed or not in the samples, which are labeled here below. And we created peptides. So this 5' and 3' peptide, I will go back here for a second, is located, right? So this is the 5' peptide, this is the 3' peptide, that's where they were located on the gene. And we wanted to see if the protein was expressed or not. So the 5' peptide, which is located before the premature stop codon, we did see protein of WBET being expressed, but when the premature stop codon was located after the 3', well, the 3' peptide is located after the premature stop codon. So we saw no protein expression there, which we were kind of expecting to see, but we wanted to confirm that the mutation in the gene was causing a truncated WBET protein. And then here I have some divergent selection analysis, looking at the Ogawa only data sets and Inaba only data sets. And I wanted to see if there was any potential selection going on. And what we do see here is not so much of a difference in the Ogawa only samples and very low divergence rates, but in Inaba we see higher rates of non-synonymous mutation, which maybe suggests some positive selection going on in the Inaba samples. So these mutations in the WBET gene, what we saw is a deletion of a C at position 305, and this is seen in all the samples that were highlighted as number 4 in the tree. And all these other mutations were not sustained, and their branches on the tree eventually died out. So some of the conclusions are a switch in serotype is caused by a mutation in the WBET gene. We know that. And it would be kind of interesting to look at and see if there's any others of the genome contributing to the switch. Another thing we want to see is the host or environmental stressor causing the switch in serotype. And I would really like to do some more in-depth comparative genomics analysis, although I want to graduate, so just want to get this done with. So going on, I also did a little project very shortly about the cholera in the Great Lakes region. So they've seen outbreaks in particularly 5 countries, Burundi, Cameroon, DRC, Ghana, and Tanzania since the 1970s. And they've reported cases of cholera every year since 1990. 3 of these countries, Burundi, DRC, and Tanzania, are located in what is the African Great Lakes region. Don't get confused if you're from the Midwest, it's not that Great Lakes. So they've had 322,000 cholera cases during 1999 to 2008. And there is a hypothesis that the spread of cholera in this Great Lakes region may be facilitated by certain local environmental factors, aquatic reservoirs.
Hey, Taylor, I think we're out of time.
Okay.
Do you want to do like a final slide and we can ask one question maybe?
Yep, sorry, I went over. All right, so I just want to say thanks to my lab. I'm sorry I went over, it's okay. That's the little project. I went over the 2 main ones. If you have any questions, let me know. All right, my name's Taylor. I have my little scans. If you want to check out my website, you can just scan. I like gifts a lot, and I wanted to thank Dr. Lee Katz and Nabil for having me talk here today. If you have any questions, let me know. Thanks, everyone.
Thank you very much. I'll ask one question, but then just due to time, we'll take it to Discord. And the question is, how much diversity do you see in the CTX phage in Haiti, I think?
So I've I haven't really looked into the CTX phage in Haiti. So I have looked into it in the DRC though.
Okay, do you want to tell us for DRC at least?
So I was seeing a lot of similarity in the DRC ones between the samples in the DRC, but saw, I believe they were ICP1 samples. So I had some reference to some regular ICP1 samples that were just on NCBI. And then I had some DRC ones, and they were supposed to all be ICP1, but I saw a huge difference between the reference ones versus the DRC ICP ones. So, but between each other, they were very similar. That makes sense.
Yeah.
But they all formed like a different tree, and I did it with the core genome. I did it with SNP. I was doing a bunch, but yeah, it was pretty crazy. But it's a huge genome. It's like 100,000 for a for a phage.
All right, thank you. You have a lot of personality in your slides and everything, and it made the talk really awesome and extra awesome, I mean. And then you have a lot more questions on Discord if you want to take the conversation there.
Yep, I will check them out. Thank you for having me.
Thank you. We'll take it.